The goal of this mini lecture is for us to understand alternative splicing and a little bit of its relationship with gene regulation. At the end of eukaryotic transcription, we have a pre-mRNA which is characterized by the presence of intron, which are the non-coding region, and exon, which are the coding region as well. So alternative splicing is a process where we get to remove all the non-coding region to remain with the exon, which are the coding one, expressing one, that are eventually going to become a mature mRNA. But what is really interesting about alternative splicing is that when we do perform the splicing, those exons get reshuffled in such a way that a specific gene or a strand of that pre-mRNA can actually give us two isoform or even more of certain proteins as well that will tend to develop during this embryonic developmental stage, or that will work around the same tissue as well. In the human genome, we see that we have a lot or many of those exon that get reshuffled, and that gives us a variety of protein that we have. This is important because when we think about it, we see that it's quite an obvious fact that the human genome is actually made of quite a large proportion of non-coding DNA or sequences. So having a process happening in the genome such as alternative splicing gives us quite a good variability of type of proteins that we can have. And when it comes to gene regulation, what we can say about alternative splicing is that in this scenario, as you can see here, you could actually have the same promoter that can be used now to transcribe a gene through alternative splicing that will actually can give two variants that can actually function in two cell types, such as your liver cells or your skeletal muscle cells as well. So having said so, in gene regulation, alternative splicing makes room or allows for a degree of adaptability to the production of the proteins that we need in specific cell types.